Hello everyone and welcome to another online seminar. For those joining our Vision Talk series, let me just quickly remind you that we are part of the worldwide Neuro initiative, a COVID-inspired seminar hosting platform for neuroscience. You will find all links and further information on these video descriptions, but I will so far encourage you to really take a look at the upcoming talks or to subscribe to the mailing list for a weekly reminder of upcoming seminars. There's usually more than one talk a day, and most previous talks are accessible on podcast platforms, like actually this very series that you can find on our own YouTube channel. Of course, hit and subscribe if you want any update. Anyway, I will be your host today. My name is Maxime, and I am part of the Baden Lab at the University of Sussex. Today, we are receiving a collaborative house, Aristides Arrenberg, from the University of Tuebingen in Germany. Ari obtained his PhD at the University of California, San Francisco, where he studied zebrafish visual, oculomotor, and cardiovascular functions. He then moved to Germany and joined the University of Freiburg, where he investigated dopaminergic neurons involved in locomotion. Finally, he started his own lab at the University of Tuebingen, where he and his team continue to work on vision and oculomotor circuits. Furthermore, they study the ecological adaptation of zebrafish brain to its habitat and behaviors. So, hello, Ari. How are you doing today?
Yeah, pretty good. Thank you very much for the invitation. So, thanks for the opportunity to present our work on motion processing in the zebrafish brain to you. We are using the zebrafish as a model for the vertebrate visual system because its brain is transparent, so we have access to every single neuron in the brain, and we work with them at a developmental stage where they're only about 4 millimeters long and show already a diverse set of visually mediated behaviors. And their brain is really tiny. It only contains about 100,000 neurons, which we hope helps us to make the brain more tractable and understand what subcortical visual processing is about. Here you can see a circuit diagram of primate cortical visual areas by Velleman and van Essen. And you can see how complex it is. And since the brain, the larval zebrafish, is smaller and it does not contain a visual cortex, we hope that it will be easier to understand its visual processing. And here you can see in yellow the 10 arborization fields, the 10 input regions in the mesencephalon and diencephalon that receive inputs from the retina that have been identified and are currently studied by others and us. Zebrafish show a diverse set of visually mediated behaviors, for example, prey capture, optomotor responses, and optokinetic responses. And these three responses will be important in the context of my talk. So let me introduce them to you. So for the optokinetic response, it's a gaze stabilization behavior where the animals try to minimize the motion slip on the retina and thereby hold the eye position stable on the stimulus. It consists of these slow phases or following phases, and then with alternating quick phases or saccades. And humans perform a similar behavior when you sit, for example, in the train and watch the trees pass by outside. Your eyes will also perform an optokinetic response. When you present a moving grating below the animal, zebrafish will show an optomotor swimming response where they try to follow the direction of the moving stripes, which you can quantify by looking at the tail beats over time. Mammals also show optomotor behavior, in particular optomotor head and neck movements. And the third behavior that will be important in the context of my talk is prey capture behavior, foraging behavior in zebrafish, where they feed on paramecia, which they hunt down. And the last stage, maybe you can see it, they have the paramecium in binocular view and use both eyes. Our laboratory is studying the sensorimotor transformations underlying these visually mediated behaviors. And we are currently mainly focusing on the sensory brain areas that receive input from the retina, so mainly the optic tectum and the pretectum, which will also be the topic of my talk today. Furthermore, we work in the premotor hindbrain, where circuits exist that stabilize the eye position, which I won't have time to talk about today. Given that the zebrafish brain is so small, it should rather make efficient use of its brain resources to be able to survive. And to make efficient use of the brain resources, the neural encoding should depend on the statistics of the sensory environment of the animal, so on the natural habitats in which these animals developed over evolution. And furthermore, we hypothesize that the circuits should be task specific. Only those information that are relevant for behavior need to be encoded. And the concept of sparseness posits that the neural activity should also be minimized in a way to save energy, having fewer neurons active at the same time, and also to make the encoding in individual neurons independent of each other. And an example of such efficient sensory encoding is found in the visual cortex of mammals, where the receptive fields of simple cells in the visual cortex look into particular positions in visual space for oriented features. So this has been shown experimentally, and you can show by simulations in the computers in neural networks that when you train a network under the assumption of sparseness and feed the network with natural images, that you get resulting units that have receptive field structures that very much look like the receptive field structures you actually find in the experiments as well, arguing that the encoding of sensory information in the visual cortex is efficient and likely adapted to the natural environments. Now, it is also known that there are visual field anisotropies across the retina. So in humans, for example, there is a point of sharpest sight, the so-called fovea, which contains a high level of photoreceptors, photoreceptor density, and also retinal ganglion cell density. And in wolves, a visual streak is present, where a higher density of photoreceptors is there in the region surrounding the horizon, so likely where the prey animals for the wolves appear. And for zebrafish, a region of increased retinal ganglion cell density has also been identified, namely in the lower temporal part of the retina, an area temporoventralis, which is not a real fovea, but a region of increased retinal ganglion cell density. And these visual field anisotropies are also relevant to the behavior of animals. Here is an example of a fiddler crab, which shows an escape response to an approaching seagull, for example. And when you present motion stimuli and measure where you need to stimulate to evoke escape responses, you find that the likelihood of the animal to show an escape response is much higher when the stimulus is presented above the horizon, but not when it's presented below the horizon. Likely because over evolution, the animal has learned that predators will not appear from out of the sand from the bottom. In related work, Ziad Hafed at the University of Tuebingen here has shown that in the primate superior colliculus, the visual field is also represented in an anisotropic fashion, where in the upper visual field, you have many neurons that have small receptive fields. And in the lower visual field, you have neurons that have larger receptive fields. And these adaptations are likely due to the different needs for processing far versus near space in the upper and lower visual field in primates. So next to these visual field anisotropies that exist, there are probably also specific processing channels that the brain forms to drive particular behaviors, and those can be anisotropic across the retina or visual space as well. Here, for example, the W3 ganglion cell in the mouse retina is present mainly in the ventral retina to look up in the sky and detect small moving items and avoid predators, and these neurons are not responding so much to larger stimuli. In the larval zebrafish, we do know the location of the retinorecipient brain areas. I've shown you these 10 different arborization fields, but we don't know very much about their functions and their differential processing of motion stimuli yet, although there are notable exceptions. From morphology studies by Robles et al., we know that different retinal ganglion cell types exist, here sorted according to the dendritic RGC morphologies, which project to different brain areas, so you can see which brain area receives what type of retinal ganglion cell inputs. In our laboratory, we have recently studied the unequal visual field sampling for different behaviors in zebrafish, and I'll show you these results in a minute. And we investigated the representation of the visual motion stimuli in different non-cortical processing channels, namely the pretectum and the optic tectum. And in the last part of my talk, I want to tell you about the integration of optic flow information across the two eyes and how the animal manages to distinguish different directions of ego motion, so that in the long run, we will hopefully get a very detailed understanding of the mechanisms underlying efficiency and robustness of motion processing. So let us start with the optokinetic response. We can place our animal inside a spherical arena. The animal is embedded in agarose, so it cannot swim away, and we remove the agarose surrounding those body parts that are still allowed to move, here, for example, surrounding the eyes. So the animal can follow the visual stimulus, and we can measure eye movements in this optokinetic response. So this is the slow phase of the optokinetic response, and we can then change the size of the stimulus. So when I say size, I mean the steradian size of the stimulus, how much of the visual field it covers here. And we can characterize how large a stimulus needs to be to drive the behavior, and where in the visual field we need to place the stimulus to drive the appropriate, to drive behavior strongest. So we can place these stimuli anywhere on the spherical surface surrounding the animal in equidistant way to characterize where the drive is best. And for the optokinetic response, we make use of a software and hardware solutions called ZebEye Track that we developed in the laboratory and for which the source code is freely available. And we use a spherical stimulus arena, which consists of 14,000 or so of LEDs, which is using much of the technology that has been developed in Drosophila vision field. And we then can stimulate in the entire or almost entire visual field of larval zebrafish, which is huge. And here you can see these optokinetic responses, sinusoidal slow phase modulations here. And these we quantify for stimuli located in different positions on the sphere. And these different positions on the sphere, we then map onto the two-dimensional space to be able to more easily look at them. So let me explain to you how this Mercator plot works, because we're going to look at these types of plots quite a bit. So we have here the azimuth running from -180 to +180 degrees. And here the elevation running from -90 to +90 degrees. So that this position here at 0, 0 corresponds to the nose of the animal. And this position here and here corresponds to the tail of the animal. This position here to the north pole or zenith of the animal. And this position here to the south pole or nidar. And we see for the optokinetic response, the color code here that shows where the response was strongest. And we see the response is strongest for stimuli that are located laterally relative to the animal and pretty much centered on the eyes. Now, we were wondering whether the photoreceptor density itself dictates whether or not the optokinetic response is good. So we mapped the optokinetic response drive in the visual field onto the photoreceptor density maps in visual space and found that the region of highest optokinetic drive, which is shown here in white, roughly coincides with the overall position of the eye, but does not very well coincide with the region of highest photoreceptor density. We were interested to know whether the visual acuity of the optokinetic response, the ability to resolve fine structures, is related to the photoreceptor spacing, so the photoreceptor density, or whether this is not the case. We performed an encoding experiment where we used stimuli of different spatial frequencies and presented these different stimuli on different locations of the visual field, and got this curve here, spatial frequency tuning curve for different visual field locations, which peaks pretty much almost at the same, always at the same location at about 0.05 cycles per degree, arguing that the photoreceptor density is not the limiting factor for the visual acuity of the optokinetic response. In another experiment, we wanted to see whether the response is mostly driven by retinal factors, or maybe also by other factors. So we inverted the animal, sitting upside down here, looking to the bottom. And we asked the same question: where in the visual field do we need to stimulate to drive the optokinetic response? And what we found is that when we invert the animal, it still prefers upper environmental positions, which correspond to ventral positions, slightly ventral positions relative to the animal, which cannot be explained by the retinal photoreceptor densities or the position of the retina. So there appear to be some extra retinal effects that determine where in the visual field you need to stimulate to drive the optokinetic response best. For the optomotor response, we mobilized animals and let the tail move freely, and then quantified the tail beats of the animal. We made use of bilaterally symmetric stimuli presenting at the same time in the left and the right side, and then use different stimulus sizes. So here a whole field stimulus was used, and in another part of the stimulus protocol we use smaller stimuli down to these very small stimuli where only two patches that are very small move into the anterior direction to drive forward motion. And to our surprise, we found that the regions that drive the optomotor response best are very different from those that drive the optokinetic response best. They are located in the temporal-ventral visual space. So there the response is strongest, and actually very small stimuli are sufficient to drive the optomotor response, arguing that it's maybe not so much a whole field driven behavior, but it's driven by local circuits here, which I think is exciting because it's has been in the past been discussed as a whole field gaze stabilization behavior. So yes, it's responsive to global optic flow and likely mediates to stabilize motion relative to the visual environment, but it only makes use of a small patch of the visual field for this. For prey capture, other authors have shown, Bolton et al., that the prey capture is performed best when the animal, when the paramecium or the rotifier is in front of the animal, which is the result of the zebrafish swimming after it. But the zebrafish also approaches it from a little bit from below, as you can see here. It's not directly in front; it's a little bit also from below. This is a side view of the animal. In dorsal view, you can see here the stimulus locations that drive prey capture best. They are in front of the animal. And these regions in the upper nasal visual field correspond very well to the regions of highest photoreceptor density, shown here for all cones or just for the UV cones, which are thought to be heavily involved in processing prey stimuli. So in summary, for these three behaviors, optokinetic response eye movements, optomotor swimming, and hunting behavior, we have shown that they all have their different peaks in the visual field where stimuli drive these responses best. The hunting behavior is driven best by the region in the retina and the visual space that is covered by the highest photoreceptor density, shown here in pink. The optokinetic response is driven best by stimuli that are centered on the retina and a little bit in the upper and nasal visual space, whereas the optomotor forward swimming is best driven by stimuli in the lower temporal visual field, a region where the photoreceptor density is already quite low, suggesting that a high level of retinal processing specificity might occur at this position. So now that we had shown these visual field anisotropies for 3 different behaviors, we wanted to relate them to the encoding in the brain. And for that, we investigated the 2 major visual brain areas, the optic tectum, which is thought to be involved in prey capture, and the area pretectalis, the pretectum, which is thought to mediate optokinetic responses and optomotor responses. The area pretectalis has its analog in the mammalian accessory optic system consisting of diencephalic and mesencephalic nuclei that process different directions of motion. The optic tectum has its homologue in the superior colliculus in mammals, which is not depicted here. And mammals have on top of that the LGN and visual cortex pathway, which is missing in fish. To look at the neural representations, we use calcium imaging and to photon imaging, the calcium indicator GCamp. You can see here responses over time to moving stimuli on the right. Every little circle here is 1 neuron, and this image is about 250 micrometers across. And we can record with the setup the brain activity at the same time as the behavior, and we can visually evoke behavior using our visual stimuli. And the first experiment is a receptive field mapping experiment where we recorded in the optic tectum and in the pretectum using stimuli that either covered the complete stimulus arena, and the stimulus arena was only presenting a stimulus to the right eye, in this case, not to both eyes, or stimuli that moved only in smaller patches of the arena down to these very small stimuli that are only 30 by 15 degrees in size. And we then record the calcium traces. 2 example neurons are shown here that respond to these gray motion stimulus phases. We find neurons like this. So this here is this neuron, which responded, shown in yellow, to the whole field stimulus, but did not respond at all to any of the small field stimuli, showing that this neuron here is a large size receptive field neuron, and it was found in the pretectum. And in contrast, we can also find neurons like this that do not respond to the whole field stimulus, but respond to very small stimuli and only for particular positions of these small stimuli. And this neuron here is then a small size selective receptive field neuron that was found in the tectum. We quantified a few thousands of these neurons and found that within the optic tectum, the small receptive field neurons prevail. 80%, more than 80% of the neurons are small receptive field neurons in the optic tectum out of the motion selective neurons. And in the pretectum, very many large size receptive field neurons can be found. Now, when we look at the distribution in the brain, we find back the textbook topography for the optic tectum. So here's a rostral region, this is the optic tectum, and they are color-coded here according to the azimuth position of the receptive field centers. Such a topographic map does not exist for the pretectum. However, we do see a certain type of map for the receptive field size distribution, which is a color code here. So the more caudal neurons tend to have larger receptive field sizes. Now, to link these neurophysiological results to the 3 behaviors that I've shown you before, we mapped the receptive field centers in these 2 different brain areas to visual space and found that in the optic tectum, the small size receptive field centers are mainly located in the upper nasal visual field, whereas in the pretectum, the large size receptive field centers, they are biased towards the lower visual field, which roughly matches the results for the optomotor responses. So let me summarize these first results. We have shown that in the optic tectum, there is a bias for the upper nasal visual field, and in the pretectum, the neurons have larger receptive fields and are biased below the horizon, which matches the visual field bias of prey capture behavior, so prey capture and tectum match, and the optomotor response drive regions, which are strongest in the lower temporal visual field, which matches roughly the locations of the large size receptive field. And for the optokinetic response, well, it's centered on the more centrally on the retina. It's not the link is not so good here yet. So now we've shown you the different visual field anisotrophies for 3 behaviors, and we would also be interested in relating the processing in the brain to the sensory environments and see how efficient the encoding is with regard to the environments in which these animals evolved. And for this, we have a collaboration with Todd Thiele, who is spearheading this project, and Emily Cooper and Scott Junti, where we recorded videos with a robot that carried a camera and moved underwater in Africa and India for 2 fish species, the zebrafish and an African cichlid, Astatopilapia botoni. And this work is ongoing. If you're interested to know more about it, then please have a look at this presentation by Todd Thiele and Emily Cooper. In our laboratory work, we noticed that optical artifacts can pose a huge problem in aquatic vision experiments. They are due to the different refractive indices of water and air, where the electronic equipment is usually placed because it does not like water. We get physical effects like total internal reflection, light refraction, light reflection, light dispersion, water meniscus, or light absorption, which can all affect the way the stimulus looks like. And to quantify this more precisely, we developed a computer graphics simulation approach in Blender, where we present a visual stimulus pattern outside the animal and then model the experimental water chamber in the computer and place a camera inside to see what the stimulus looks at the position where the fish eye would be. And we have developed also in practice a water container that is spherical and made out of glass, which we think is much superior to many previously used water containers. So let me highlight one result here we have. So here we can see the stimulus, how it looks like from within the water container. Looks very clean and clear. And from within the Petri dish, you see all these optical artifacts, which are mostly resulting from total internal reflection that happens at the water surface, but also at the Petri dish bottom, at the interface between the plastic and the air. And we think that our spherical glass bulb arena is an improved setup. And we also showed this experimentally in this experiment, where we measured direction selectivity. So we have a stimulus that moves in 8 different directions, and we measure the neuronal responses. This is the response of a single neuron here. This neuron is direction selective. So we plot these responses in the polar plot. We see that it prefers the direction 90 degrees, this neuron. And we can then measure hundreds of neurons in the optic tectum for both a setting where we use a Petri dish lid as a container or a glass bulb to see which preferred directions prevail. And we find that when we use the good glass bulb, we find 4 represented preferred directions in the optic tectum. However, when we use the Petri dish lid, we only find the horizontal directions, 2 directions, but not the vertical directions. They are missing. So why is that? We think we can fully explain this by the total internal reflection that happens in the Petri dish lid, because these reflections will lead to inversion of the vertical stimulus. You can see these arrows point in both directions, and the letter L is inverted. Whereas for horizontal motion, the stimulus is reflected, yes, but it does not change its direction. The orientation of the small leg of the letter L is still pointing to the right. So only the vertical direction is affected, but not the horizontal direction. With these direction selectivity experiments, we have found that both in the pretectum and in the tectum, the same 4 directions are represented, which roughly correspond to the horizontal directions and the vertical directions. Now, how can the animal make use of these different directions that it has encoded in its brain to judge ego motion? This is not a trivial task, because the animal needs to decide whether to move its eyes or to swim. When the stimulus is rotating, it can best control or stabilize the stimulus by just rotating its eyes. for which the pretectum needs to activate via different processing steps the abducens nucleus and the oculomotor nucleus to drive the extraocular eye muscles. Whereas for the optomotor response, the pretectum needs to relay the information to the reticulospinal system, to the nuc MLF and to the reticulospinal cells in the hindbrain, which then drive the muscles in the tail to drive swimming. And this is mainly shown optomotor forward swimming in response to a forward moving stimulus. So how can the animal distinguish these two behaviors, or make the decision for these two behaviors given the optic flow that is present? It likely needs to decompose the rotational components from the translational components for this task. And our laboratory has shown in the past that neurons exist that are selective for translation in the horizontal direction, and more recently also translation selective neurons that are present for encoding vertical directions. And we also found rotationally selective neurons, so that we have come to the conclusion that the most likely common circuit motif is such that direction selective cells in the retina project to the other brain half, because the optic chiasm is completely crossed, and activate their monocular direction selective neurons in the pretectum, shown here in green. And then already within the pretectum, binocular selective neurons exist that receive input from two such neurons that have connections to both eyes to make a binocular neuron. And this neuron can then obtain a translation selectivity or rotation selectivity. And in our data sets, we find every single one of the 6 possible degrees of freedom for rotation and translation. We found it to be encoded. So we found neurons for it. And these results are in contrast to recent findings by the Naumann group, by Naumann et al., who claim that the selectivity for forward-moving stimuli is not present in the pretectum, and that the selectivity for sideward-moving stimuli is also not present in the pretectum. So in their model, they need processing that we would claim is visual processing, namely to compare between the two eyes to decide whether a stimulus is moving forward or moving sideward. But this decision is not happening in the pretectum, but that much more of the brain, the early and late anterior hindbrain, are needed for this. Whereas we think that the visual processing is occurring within the canonical visual pathways, and likely no further processing is needed. So they could, in principle at least, directly inform optomotor and optokinetic behavior. So now that we have these binocular selectivity data, and we wonder how do the receptive fields of these neurons actually look like, there's beautiful data from fly research that shows that receptive fields such as these exist in the tangential cells of the lobular plate in flies, where the neuron with its optic flow directional selectivities is really selective to the type of global flow that occurs during a particular rotation direction. So here we have rotation along the body axis. This is roll, and this is the resulting global flow. And then this neuron here directly forms a template for this type of motion and can be used to judge rotation versus translation. These beautiful experiments have been done using electrophysiology, which is difficult to do in zebrafish. So to do something similar in zebrafish, we first had to develop a new stimulus protocol that works for calcium imaging, which is slower, but with which you can record hundreds of neurons at the same time. And we succeeded in doing so. We call the stimulus protocol the contiguous motion noise stimulus, where the motion at any given pixel position is correlated in space and time locally. And we can then see the resulting brain activity and perform reverse correlation to ask how did the stimulus look like when the neuron fired. And this allows us to estimate the receptive fields of the neurons. And in the following slide, I show you 4 example neurons in the estimated receptive fields. So these 2 neurons are unimodal, and these 2 neurons are bimodal. So the unimodal neurons have a single patch in their receptive field where they prefer a certain array of directions. And the bimodal neurons, they have 2 modes. And in this case, for example, it's a translation neuron that prefers translation with this here as the point of expansion. When we look at the unimodal neurons, of which we find very many, this is their receptive field center distributions, distribution in visual space. We find back the same directional tuning for the preferred directions. So these neurons are maybe not the most interesting ones, but these bimodal ones, they are really interesting. We find them mainly in the pretectum. And for those, we can look at the mode distance, so the distance between two modes of the same neuron. So for example, I don't know, this neuron here has its one mode here and its other mode here connected by the lines. Every pair here is one neuron, and the mode distance is not uniform. It can be clustered with a clustering algorithm and according to whether or not the neuron is monocular, so where both modes are contained in one eye, or binocular, into 3 groups of bimodal neurons. So these neurons here are the monocular neurons, where both modes lie in the same eye. And these 2 are binocular neurons, either with a smaller distance or with a larger distance. And we can also ask how these different modes, how they combine the directions. Here we sort the bimodal neurons according to their direction selectivities of the right mode, so what the right eye likes to see. And we see here that in the when in the right eye the stimulus goes to the left, in the left eye the stimuli should go to the right, making it translation selective. And when we summarize these data into these polar plots, we find that for vertical motion, neurons exist that are selective to translation, and also neurons that are selective to rotation. Whereas for the horizontal motion, we mainly find neurons selective for translation, not so much for rotation. So neurons that like forward motion or backward motion, but not so much clockwise or counterclockwise rotation. And to really understand how the neurons are capable of helping the animal to judge a particular ego motion direction, we can now relate the receptive fields that we measured to the closest translational and rotational fields to ask how similar the receptive field is to one of these global flow trajectories. In this example here, you can see a neuron with its receptive field in red, and in the back in gray, you can see its closest global optic flow field. And you can see that this neuron likes translation, apparently, whereas this neuron down here likes rotation about this point. So we find that the responses of the receptive field generally match very well one of these global flow directions. And we are currently investigating how well it really helps the animal to make the distinction. One thing we are quantifying for that in this ongoing work is the similarity of this stimuli to rotation and translation. We find a lot of neurons, these gray ones here, which can be modeled by either rotation or translation, and we find other neurons, these ones here, which are translation selective. There's no global flow field for rotation that would match well for the receptive field of the neurons, and other neurons that seem to be more rotation sensitive, where we cannot find a translation global flow that would match very well with a neuron. So in summary of the second and last part of the talk, I've shown you that in the brain, in the pretectum and optic tectum, there's a representation of 4 orthogonally arranged preferred directions. And I've shown you this new contiguous motion noise stimulus that we use to precisely map the flow fields of the neurons and find out how they really process rotation and translation, help the animal to make decision in mixed environments, as well, where rotation and translation are mixed. And I've shown you evidence for binocular selective neurons and how they could, in principle, inform optomotor and optokinetic behavior. Of course, it still needs to be shown what path is really chosen in the fish for driving optomotor and optokinetic responses. That is still unknown. So with that, I would like to thank you for your attention. I would like to thank our sponsors. And I would like to thank the PhD students and scientists in the laboratory who performed the work I presented today: Kun Wang, Yue Zhang, Florian Demelt, Rebecca Meyer, Julian Hinz, and Tim Platnik. So yeah, thanks again. I'm open to any questions you might have.
Thanks a lot, Harry. That was very impressive. I would ask everybody in the audience to join us in a room if you want to ask yourself questions to our guest today, or if you just want to join us for further talking about this topic. So Ari, I have a question in the chat for you. A question from Tom Baden, who's asking, how do you think we can link the 4 to 90 degrees of direction selectivity axis you see in the spherical arena to the 3 to 120 degrees separated direction found previously?
Yeah, from the retina, you mean? Yeah, so that's a good question. So we have in the retina 3 directions encoded. In the tectum and pretectum, we have 4 directions encoded. And yeah, it's really a question, why do the animals do that? And I mean, there's a publication by the Martin Meyer lab that claims that at least how in principle you could obtain these 4 directions of responses by neurons in the tectum, that syn neurons that would help in establishing these new response types. So that's at least an explanation, like how you can get these responses, but it's still unclear how the animal actually makes use of it. So why it's better to have this one representation in the retina versus the other representation in the brain. And yeah, right now I don't have a very good explanation to it. I mean, for the vertical directions, it has been proposed that the orientations, I mean the rotation orientations that are encoded, that they match with the axes of the semicircular canals in the vestibular system, and also with the extraocular eye muscles to make it more efficient, the encoding. So it's maybe something you need at the level of the tectum and pretectum, but maybe not so much yet at the level of the retina. Yeah, but still an open question, very interesting one.
Very interesting. Sorry, stupid personal question. Will you consider spectral stimulation to add to your motion? You showed something between the UV density, so UV cone densities, and the distribution of some receptiveness of motions. So
yeah, so far we have not been able to investigate that because our stimulus arena is monochromatic. It's green to us. Activates mainly the red photoreceptors of the fish. And that's something we would also be interested in the future in an effort to understand how color processing and motion processing are disentangled, and to what extent it goes together and to what extent it's different. I mean, it has been shown that for the optomotor response, it's more strongly driven by red and green stimuli, not so much by blue stimuli. For UV stimuli, the data is still lacking, and it will be interesting to find out what exactly these dependencies are. And it's yeah, something that I can only speculate about right now.
Mm-hmm. So do we have more question? What I'm looking for, can I just ask you what is the maximal resolution you can achieve with this simulator and your spherical arena?
Yeah, so I said it's about 14,000, a little bit more than 14,000 LEDs. So along the horizon, the azimuth, where the spacing of the LEDs is optimal, there we obtain about a degree per LED pair. So it's like yeah, about the resolution limit of the retina. And in other parts, it's maybe a little bit worse. So yeah, it's on the order of one LED per photoreceptor, but it's yeah, if you want to look at prey capture behavior, so then it's maybe better to use an arena that has a still a little bit higher resolution than what we do. I mean, we right now are mainly interested in the global flow processing for the optokinetic and optomotor responses. There, it's not so important to have a very spatially highly resolved stimulus.
For the average. Thanks. I have a follow-up question from Tom Baden, who's asking, how are these DS cells linked to the optic flow response you showed?
How are they linked to the optic flow response? So it is known that the DS cells in the pretectum are responsible for driving optokinetic responses. I mean, that's data also from other species, rabbit and other mammals, that show that you can that this region is sufficient and necessary for driving optokinetic responses. And also Fumikubo has performed experiments with optogenetics in zebrafish to show that you can activate the pretectum and drive the optokinetic response. For the optomotor response, this is not so well. Well, it is also thought that it's mediated by the pretectum, but the direct evidence is maybe not as good yet. So arguing that the direction selective neurons in the pretectum are responsible for mediating these behaviors. Now, that would mean that the responses in the tectum that we see have mostly modulatory function. I mean, that's data that if you ablate the optic tectum, then you actually don't get a, then it's not completely absent, the optokinetic response. So that will be my take on it, that there's a modulation, but we don't know very well. Also within the pretectum, and there's like several pretectal areas, and we don't know which ones are really driving it. So I think for that we need to do like single cell ablations and similar things to see what each neuron really does.
Thank you for that. I have a comment from Advik Bayer. Hi, Ari, great talk. A neuron can have a large receptor field, but still be selective for small size stimuli. Maybe I missed it. Did you find such neuron?
Yes, we looked, we found such neurons that have a large receptive field, but really preferred small stimuli. In part of the pretectum, but they were a little bit difficult to grasp in terms of the reproducibility. So in some recordings, we found many of them, and they would be really excited because they might be like neurons that alert the animal to, oh, look, there's like a prey item swimming around, and that would be really cool. But in other in animals, we did not find so many of those neurons. So for that reason, I did not include that data today, because we still need to do further work to really understand what these neurons are doing better. The other way around is very clear, like neurons that have a small excitatory receptive field, but that are inhibited by large field stimuli. I've shown you like this one small size neuron in my talk that I called small size selective neuron, because it is suppressed by stimuli that are larger. And 80% of the motion sensitive neurons in the optic tectum that are small size responsive are also small size selective, meaning that this is a general concept that the tectum will not get activated very much by stimuli that are global flow stimuli that move everywhere.
I have a comment from Michael Reiser, but I see it here with us in a room. Maybe Michael, you want to ask a question yourself?
Sure. Can you hear me?
Yes.
Okay, great. I actually have two questions. It was a really great talk, very thought-provoking. So one is about the optic flow structure, the fields you find. They seem primarily to be centered close to the equator, if I understood correctly. And I wasn't sure if that was more about that particular stimulus and that visual display system, or if you think this is like fundamental for the neurons, is one question. And the follow-up question is just about speed tuning. Do you also, when you do all these mappings, are you following through something like temporal frequency or speed tuning, you know, from the directionally selective neurons through the flow fields to the behavior? Because in flies, we have big mismatches there. Okay.
Yeah, for the first question, we don't think as of yet that this is necessarily really like what the animal only cares about, that they are like all oriented along the horizon, although we think it would also make sense, but we have to be cautious there, because I maybe glanced over it in my talk, but for the receptive field mapping that we performed, we still use a cylindrical arena, which only covers the visual space from -40 to +40 degrees in elevation. We don't have our full globe setup yet present, and we would like to repeat the experiments with the globe setup to really see whether this arrangement, where it's mainly where the bimodal neurons are mainly located along the horizon, whether this really holds or not. But it looks from the data like, yes, they care more about this translation and rotation that takes information from those regions of space. But yeah, that's something that we need to look into more detail. For the other question, the speed tuning, a very interesting question, and one that is difficult to answer with our current data. Unfortunately, we do have a stimulus that has a certain speed statistic, and when we draw the stimulus arrow, like the size of the arrow, it looks like, well, maybe it's representing speed, but it's not really speed. It's more like how much of the stimuli were present that went in this direction when the neuron fired. So it's not really only about speed. It's a mixture probably of a speed tuning and how direction selective this neuron is. And that's something where we need new experiments to test precisely the speed tuning, so we can really nail it down and find out what these neurons really like. And this is an important question also in the concept of mixed optic flow regimes, where you have rotation and translation mixed, and you have like speeds and of certain size, like how well the animal is really able to distinguish those will also depend on the speed tuning. So to really nail it down, we will have to understand that as well. Thanks.
Thank you.
I'm sorry. Do we have any more questions? Give me a second. Yes, I have one from Keisuke Yonehara. It's a great talk. Is it possible that retina-independent mechanisms may generate direction selectivity in relevant brain areas? Or do you have evidence that these are mediated by retinal DS cells?
Yeah, so we think that the most parsimonious explanation is that the direction selectivity already exists in the retina, and it does not need to be established de novo in the optic tectum or pretectum. However, there's also evidence that it is actually possible to re-establish direction selectivity in the tectum and the tectum independent of the retina. So those are experiments performed in Florian Engert's laboratory, where they had a stimulus where they forced the neurons to rewire to the brain in such a way that the retinal ganglion cells from both eyes would end up in one tactile half, but not in the other. And then they presented stimuli to one eye and the other in such a succession that for each eye it was not a motion stimulus, but if you put the two eyes on top of each other, then there was a motion stimulus, and they found neurons in the optic tectum that would be selective for that. So arguing that yes, novel direction selectivity can be established in the tectum, but if you ask me, I would think that this is probably, since we already have it for free in the retina, why should the brain not use it? I would think that they probably, the tactile neurons make massive use of the direction selectivities in the retina. But we need more data to really say. But that would be my take on it.
That's very interesting. Okay, well, thank you, everyone. I will soon end the live streaming. So if people want to join us, I will suggest they do now, because I will close the link in a few seconds. Thanks again, Harry, for accepting our invitation and talk to us today. And for the audience, we will have another talk in 2 weeks' time. So hope to see you there. Bye. And the live stream is off. Okay.